From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on September 5th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 107th episode of Matters Microbial. Many thanks to every listener, viewer, guest, and supporter. Keep those likes, listens, and views coming. I enjoy doing this podcast, and I hope you enjoy it too. It really matters. This has been the first week of my final class of microbiology here at the University of Puget Sound. My students, Micronauts, are wonderful. Here they are in class displaying some of the microbial mementos I have given them. And here they are in lab. I truly appreciate their enthusiasm, patience, and willingness to collaborate with me in both lab and lecture. I will miss them. Here is the extensive amount of microbial swag that I handed to all 19 students on the first day of class. It includes some microbially themed candy seen here. And they have mottos of my class: Microbes Rule, Quality Quorum, First Evolved, and last extinct. I also enjoy using photobacterium, which is bioluminescent, to teach basic microbial skills. For one thing, it makes me happy to show up early the next day to see the Micronauts plates glowing in the dark, seen here. The students did a wonderful job streaking a plate to single colonies, seen here. Also, making beautiful dilution plates, seen here, that can look like stars in the night sky. As I've said before, I like to have Micronauts explore their reusable water bottles to see what kinds of microbes reside there, and this is independent of most forms of cleaning, by the way. I try to normalize this process by calling the microbes water bottle buddies, as you can see from this course sticker designed by Ethan Kokek. Here is a swab from the inside wall of a 1 Micronauts water bottle, which shows a very high bioload, which may not be a problem. When this same Micronaut streaked out from the swab, we observed some brightly fluorescent, not bioluminescent colonies seen here. We hypothesized that the glow is due to some organisms making something called siderophores, compounds that scavenge iron, an adaptation to low nutrient conditions. We hope to find out. Finally, I performed a demonstration of ice nucleation activity by Pseudomonas syringae. I added some of that culture to very cold water, and you can see how the bacterium causes ice to form in this short video. I'll put links to this phenomenon in the show notes. For my Micronauts, I remind them that the shape of the ice nucleation protein made by the bacterium is similar to the shape of water ice crystals. This seeds the formation of ice. This also tends to pop plant leaf surfaces where this bacterium lives, leaking and providing nutrients to the bacteria. Microbial tip of the hat, as always, to Dr. Joe Handelsman of the University of Wisconsin-Madison, who taught me this procedure many years ago. Relevant to today's topic, I gave my Micronauts their first assignment. Each student received a plush microbe keychain, like this one for Listeria, seen here. I had each student then create a "Hello, I'm" style name tag with important facts about their bacterium. This Micronaut did it for Listeria. It's really quite impressive. What I like best about this assignment is that what they put on the name tag can be shared with non-science folk, because science communication is extremely important. Today's topic is about a bacterium and a disease, and the topic of food safety. Listeria, listeriosis, and how we can protect ourselves from the latter. Scanning the internet, there have been many outbreaks of listeriosis over the years. So I am honored to welcome a wonderful friend and colleague to today's podcast who knows quite a bit about Listeria. Dr. Lisa Gorski, recently retired from the USDA and currently a consultant on food safety issues. Welcome to the Quality Quorum today, Lisa. It's so wonderful to see you.
It's great to be on, Mark. I'm very happy to be here.
Oh, thank you so much for this. Would you mind terribly telling everyone the origin of the name Listeria?
It was named after Joseph Lister.
Joseph, can I say father these days? The parent of antiseptic surgery, and really a wonderful figure in the history of microbiology. I don't believe it has anything to do with Listerine, though.
Well, that might have been named after him too, but
yeah. So in any event, this is an interesting organism. It has all kinds of fascinating things. I talked about it a little bit in the class that I'm teaching, but I think the best thing to do, if you wouldn't mind, is give a thumbnail sketch of Listeria and listeriosis.
Okay. So most people these days know Listeria because it causes a foodborne illness called listeriosis. In numbers of cases of listeriosis in the United States per year, it's not very high. So you maybe get a few hundred to a few thousand cases a year. The issue with Listeria is its fatality rate. So it can be from 20 to 25% fatality rate. And beyond that, just as an example, in 2019, there were only 134 cases, but 98% of those resulted in hospitalizations, and 16% were fatal. So it's a very severe illness for people who are susceptible. And who are susceptible? Immunocompromised people, the very young, the very old, ill people, people who have immunocompromising diseases, and pregnant people, especially the neonates in pregnant people. It can cause spontaneous abortion. So in that case, it's heavily surveyed for by companies and by Food Safety Inspection Service. It's kind of the poster child for cell-mediated immunity because it gets into people by ingestion of food, and then it immediately can become internalized into phagocytic and non-phagocytic cells. And it manages to hide from the immune system because it hides inside animal cells. And it can propagate itself inside those cells, and it can move between them actually by just butting up against the cell next to it and moving over into it. It's got a wonderfully interesting way of moving through human cells by nucleating the host cytoskeletal actin. And it basically polymerizes actin and uses it as a means of propulsion. And it makes these comets inside the cell. There are wonderful videos of it, which I'm hoping you can link to.
So I wanted to say for most people that it's a really interesting organism. I did some reading about it because I teach about it a little bit every year. And one of the things that fascinated me, maybe you can address, is that in many cases, most cases, Listeria is dealt with inside the gastrointestinal tract with no trouble. So that's external to the cells. Right. But a small subset will invade the cells, and there they're protected from the immune system.
Yes.
And so, in fact, I think we have a video of how these actin rockets, and I want to remind listeners and viewers that this video, if you're seeing it, has been sped up. This is not how they truck about normally in real time. And I also heard, Lisa, something fascinating, that when they invade cells and they basically modify part of their teichoic acid so that they're recognized by a macrophage or a phagocyte, and then they get internalized the way that our white blood cells act, which are kind of like tame amoebae.
Right.
And you think, oh, that's okay. They're going to get fused to a lysosome and then get digested. But they pop the lysosome.
They pop the lysosome.
Yeah.
So they pop the lysosome, and then they can multiply inside those cells. And as far as virulence goes, as disease progression goes, it can then make its way to the liver, the lymph nodes, the spleen. It can cross the blood-brain barrier. It can cause meningitis. It can cross the placenta, and hence the miscarriages that occur.
Lisa, you mentioned certain numbers of fatalities due to listeriosis, and I was curious, can you give me some idea of how that's occurring? Overreaction of the immune system, cells bursting apart?
Well, what happens is when it becomes systemic, and it can find itself in certain organs of the body. If it infects the liver, it can lead to liver failure. So the organ itself goes down. Once it crosses the blood-brain barrier and you get meningitis, then it's just classic meningitis symptoms, the swelling of the brain and the spinal column area that would cause issues. And once it crosses the placental barrier, just growth of the fetus, which would cause spontaneous abortion. So it really depends on where it finds itself in the body afterward. And it's usually just a failure of those organs to function. Healthy people, if you get a significant enough whopping dose, you might just get normal case of foodborne illness. You might throw up but and you may not even report that. You just think, oh, I just got sick from something I ate. But it's the nasty ones that are really an issue.
By the way, if you don't mind my asking, because we don't often have people who talk about diseases on the podcast, how are these things reported?
Well, in the case of listeriosis, it usually pops up a few weeks after ingestion, and the person gets so sick that they're usually hospitalized. And so in that case, for the other bacteria and viruses that cause less severe foodborne illness, a lot of them are not reported. You get reporting of some people who will go to their doctor because they've become acutely ill with something. But whenever you read statistics, there's always a caveat in there that a large proportion of foodborne illness may never be reported and may never have identified the etiological agent.
And so one of the most important things that a lot of agencies do, including your former agency, is try to monitor the prevalence of these possibly disease-causing organisms. It doesn't mean necessarily that that's a reflection of disease cases, although you get called in when there's an outbreak.
Yes.
So what kinds of foods have been impacted by Listeria?
So Listeria most often is found in ready-to-eat products. Sometimes ready-to-eat meat. And in fact, some people, when they are pregnant, are told not to eat deli meats. And sushi and raw fish. And those are because of possible Listeria contamination, as well as mercury contamination for the fish. But so seafood, ready-to-eat meats, dairy, produce, mushrooms, it basically will run the gamut. Listeria itself, when it's not being a pathogen, is ubiquitous. It's present in nature. It lives out in nature decaying plant material. It's a saprophyte. So it's very commonly found out in nature. It's carried by wildlife. And so it's easy to find in agricultural environments and as well as in food processing environments, because once it comes into the food processing plant, it's a really hardy organism. It can grow at very low temperatures, and I mean grow. It will multiply in the refrigerator, which is an issue for foodborne illness. And so food processing plants are always on the lookout for potential Listeria contamination, because if you've got one, you've got millions. It will just grow. And in fact, very early enrichments for Listeria took advantage of that. They did enrichments in the refrigerator at 4 degrees.
That's fascinating to do the enrichments that way. Is this another source of concern with sprouts, or do I have sprouts on the brain?
There have been cases of sprout contamination with Listeria. There are other bacteria which are more commonly associated with foodborne illness with sprouts, usually Salmonella, some E. coli. But Listeria has caused some. Sprouts have been a really good model system for us to just look at Listeria contamination of plants in general. But yeah.
So further than that, I know certain types of soft cheeses are vulnerable to contamination. Is
Yes.
that because they're not pasteurized in some cases, like homemade, for example?
Non-pasteurized cheeses can be an issue. If they're made correctly, they're usually not.
Yeah.
So the soft cheeses tend to be implicated more than the harder cheeses, but pretty much I've seen Listeria linked to almost everything.
Yeah.
And
And this is an important concept for listeners and viewers to understand. And I say this not to be flippant. It's kind of like the pig pen idea from Charles Schultz. Remember how he was surrounded by dust
Yeah.
and dirt? Well, we're surrounded by microbes all the time. We're swimming in a sea of microbes. I know, for example, because I've had friends who've done it, Lisa, if you swab your skin, you can find MRSA there, but you don't have a disease. I guess it's in about 60, 70% of people that they were testing. So to me, it almost seems like it's about ecology more than anything else.
It's definitely about ecology. All of it is ecology. Even the disease state is an ecology. One of the things that you look in properly pasteurized foods and in properly handled foods, all foods, unless they're sterilized, should have a natural microbiota to them.
Yeah.
Definitely raw produce does. And that's good, because if you've got innocuous microbes there doing what they're normally doing, they're going to block sites for the pathogens to hook onto. Mm-hmm. Or they're just going to make it more difficult for the pathogen to be there, or they're going to just outgrow them. They're going to outcompete them. So if you get a Listeria or a Salmonella or an E. coli hopping onto a lettuce leaf, for example, the other bacteria that are there might just kick it out or outcompete it, because it's not a natural environment for the Listeria or the pathogen.
I don't believe I've told you this, but when we've surveyed what we see on the inside of water bottles, we will occasionally find microbes that produce antimicrobial agents. And so they're duking it out for space. I guess the fancy term is niche exclusion, right? And that's what we're dealing with here. What I wanted listeners and viewers to understand is there's this whole PR business we have in microbiology about devil microbes. They're always bad. They're germs, that kind of stuff. And I really go on rants about that in class, which I won't do here. But I want people who are listening and viewing this to understand it is a microbial world. We just live in it. And those few cases that they cause problems are actually the exceptions, not the rules. So there is a lot of hysteria about Listeria.
Definitely.
Yeah, I need Def Leppard to sing that song, right? Believe me, I've thought about it. So with that in mind, we've shown a little bit about actin rockets. We've talked a little bit about the mechanism that Listeria has to live inside the cell. Fascinating subject to have an organism adapt to life inside one of our cells. Just to remind listeners and viewers to avoid the immune system, I guess those extensions are called philopodia, where they go from one cell to another without being released to the bloodstream.
Right.
It's fascinating. But now we move into the issue of how we make sure that our food is safer. I won't say safe, right?
Right.
And again, you brought up the idea that a lot of prepackaged food, this is going to happen because we often don't think about the process of preparing our food that we go to fancy supermarkets or to get, right? We don't
Correct.
understand a bagged salad has to be put together by people.
Correct. Well, a bagged salad is processing the produce in a processing plant, and usually requires requires, just by virtue of the processing, damage to the produce. Um, when you damage the produce, you get leakage of nutrients, and you make an environment which is conducive to growth of many microbes, not just the ones that are naturally there, but ones that might just hitch on for a ride, such as a foodborne pathogen. And those bagged lettuces and such are usually washed in vats where they're all together. So if you have one piece of lettuce which has a quote unquote bad microbe on it, it can disseminate across the whole bath, as opposed to picking up a head of lettuce, an individual head of lettuce, which was taken from a field, which you are then processing yourself in home. It's less likely to have those contamination issues.
So I guess a really important question is, how do you detect, in this case, Listeria? And there must have been old school ways, and I know you've worked on new school ways. I spelled school SKOOL for you.
Thank you.
It's so good to see you, Lisa.
I know, it's been a while. So yeah, the old enrichments used to do 4-degree enrichments. That's not feasible in today's world. Yeah. So there are standard enrichment media, various different kinds of enrichment media that take advantage of physiology of Listeria and carbon sources that it uses. It is a gram-positive organism, if we haven't said that already. So there are some dyes that this organism is resistant to that will inhibit the growth of other organisms. So some of those media have these dyes in them. They also take advantage of the fact that Listeria can grow at different pHs and at various temperatures. So enrichments usually happen at 30 degrees instead of 37 degrees, and they're multi-tiered. So there will be an initial pre-enrichment and then a subculture from that into various more selective media. So in the classical phenotypic, just doing microbiological isolations, it can take a few days from your first sampling to a solid colony, which you're going to then now say, look more closely and see if this is Listeria monocytogenes. And it's usually monocytogenes, which is the species that we're looking for. There are molecular methods which can take those pre-enrichments and take a portion of that and use multiplex PCR or even immunoassays to say Listeria positive or negative. Now, Listeria has 17 different species in it. Only maybe two that are definitely pathogens, although there have been reports recently of a recently identified species that might have pathogenic tendencies in humans. But most of those others are not pathogens, and they're ubiquitous. They're even more ubiquitous than the monocytogenes. But the monocytogenes is what they look for, so is what they're worried about. They look for Listeria species because they figure if they've got Listeria in their food or in their processing plant, they can put a hold on it and block it from going out or put out a recall. So the quicker you can identify that, it saves lives, it saves money, it saves everybody a whole lot of time.
We're just going to call that win-win-win.
Yes.
I wanted to tell listeners and viewers, we had a previous guest on this podcast, Dr. Arturo Casadevall, and he actually doesn't like the term pathogen that we all use. Me too. And the reason he doesn't is he points out something you're alluding to, Lisa, that there are some microbes that are completely do not cause disease. In some circumstance, they do in others. The example that I like to use in my class is you've got bacteria in your gut that make vitamins that you need. if you get physically hurt, say a car crash or something similar, you release those bacteria into the peritoneal cavity, you'll get sepsis. So in some sense, the ability to cause disease, what we call pathogenesis, is a matter of context and location. Kind of like a realtor says it's location, location, location. It's context, context, context. So this idea of having things that could potentially cause disease out there is probably more accurate than we know. If you compare the barcode that we use to identify microbes between Bacillus thuringiensis that makes an insecticide versus Bacillus anthracis that causes anthrax, those barcodes are practically identical.
Yes.
So there's a lot of diversity out there is the point. So how do you define whether or not you have something that can cause disease, or do you not take a chance? That's the issue.
Generally, when they look for Listeria species, it's because they're not taking a chance. They're just, and this is important because not all Listeria are created equal. And I'm talking not all Listeria species and not all Listeria monocytogeny strains. What I have found in spending many, many years delving into the physiology of single organisms is a huge diversity at the species level. So, for example, about 50% of the Listeria monocytogenies that are isolated from food processing plants are no longer pathogenic. They have acquired mutations in virulence genes, which make them highly, highly reduced in virulence. And there are specific virulence genes that are more common, that this happens to more commonly to. Internalin A, which is the protein on Listeria, which does the initial attachment to epithelial cells, gets truncated and then is no longer functional. Now, when we've looked at environmental Listeria, we find this is much less the case. At least in the areas where I've done a lot of my surveys in California, the predominant number of Listeria monocytogenies we isolate from nature, from water, from the environment, have the full complement of virulence genes, and they are intact. And beyond that, strains can have different groups of virulence genes. There are 3 pathogenicity islands in Listeria, 3 groups of genes which cause varying levels of disease. Pathogenicity island 1 is your basic subset, the things that get you hooked onto the cell, things that get you inside the cell, and things that get you moving on to another cell. But there are additional virulence islands in some Listeria strains which cause even more severe disease. So the people who get like the severe meningitis, the neurological issues from Listeria, those strains tend to have up to three pathogenicity islands, which have more stuff in them that causes more severe illness. It makes them able to get past different issues in the human immune system. So, all of that saying, not all Listeria are created equal. And within the Listeria species, there are multiple subtypes, and the ecology, not only in the processing plant, but out in nature. Not to mention, there are some listeriosis outbreaks that happen where multiple strains are isolated from the food source, but only one strain is isolated from the person. The person is also doing a selection.
Yes. So I have this, go ahead. I wanted to say, Lisa, I have this expression, and it actually annoyed one viewer, and I really took it to heart. The hand of Darwin is on everything that lives. And so evolution is taking place at all times, no matter what. Now, what was so fascinating to me as you talked to me about this is that in the early days of microbiology, the idea of attenuating something that caused disease by growing it at a temperature different than our body temperature, so the organism mutationally adapted to living at that temperature, and many of them could no longer cause disease.
Right.
That can work in the other direction, as you just said, where the human, those few that are able to occupy humans and spread themselves about will win, as it were.
Yeah.
Although it's not really a race, is it?
No, it's just nature.
But exactly. And it's like, and that's the one thing that makes it tough to teach microbiology is the level of diversity is there. Now, I happen to know that your PhD advisor was with me 100% on this subject.
Yes,
because he taught me. All right. But it's really 100% true that we are, I want to imagine fingers pointing at one another, the microbial world and the human world. We're interactive all the time. And we tend to think it's one way, and it isn't. And that's a great example. I will also put links to pathogenicity islands, which are often a sign of, I shouldn't call them just pathogenicity islands. I'll call them genomic islands. Sounds like a resort. But what I want to tell people is that that's often evidence of horizontal gene transfer.
Yes.
That is to say, they're shuffling these things around again, which makes this a wonderful system for you to look at. When I say wonderful, I don't mean in terms of disease.
No, but fascinating.
Yes. Yes, I think we learn a lot. By the way, for listeners and viewers, just to backtrack a minute, there was the late Elio Schechter used an expression with me quite often called cellular microbiology, which doesn't mean the cell biology of microbes. What he means is we understand our cells better because of the way that microbes interact with us. And actin rockets are a really great example of this. It taught us a lot about how the cytoskeleton works in us normally. So even disease-causing organisms can be useful.
Well, actually, there have been studies of using attenuated Listeria monocytogeny strains to deliver medication to targeted cells in people. They've as a vaccine vector because it can get into those cells. And if you manage to make it not cause disease, then maybe you can use it for something productive.
Exactly so. Exactly so. The idea of popping a membrane inside a cell that has a membrane continues to fascinate me.
Yeah.
It's kind of like taking an egg out of its eggshell without breaking the eggshell.
That's true. That's a good analogy.
So I think what people would love to hear about is the process of when you did some of this work, what people would send you and how you would analyze them. You've mentioned before that there are ways you can use what's called multiplex PCR, where you look for several target genes in a sample. Presumably some are due to Listeria genes. The other are immunoassays, which will be specific to some surface component of Listeria. So they send you samples. Is that how this would work?
So what we did, the work that we did in my lab tended to focus on, we would use actual disease-causing strains to study how these organisms would interact with plants, and well, specifically produce, because I worked for the U.S. Department of Agriculture in the Produce Safety and Microbiology Research Unit. So we were focused on produce, but because Listeria was ubiquitous in a lot of different areas, I kind of collaborated with people in different, such as poultry and things like that. But so we would do individual experiments on plants with isolated strains, but we also did a long-term 5-year longitudinal survey of surface waters along the Central California coast, ponds, rivers, streams, lakes. And the idea was we just wanted to see what foodborne pathogens were out there in nature near agricultural regions. These waters were not necessarily used for irrigation, but they were useful. They were close by for wildlife who could traverse the water and then get into the agricultural fields, which can be a problem. And they were available if there was ever flooding. So sometimes they would flood the fields. And so different people in my unit looked for E. coli, S-tech E. coli, Shiga toxin producing E. coli. I had a separate project where we looked at salmonella diversity, but we also looked at Listeria monocytogenes diversity. And so for 5 years, we processed water samples, basically a Moore swab. We take a piece of cheesecloth, ball it up, and toss it out into the water, leave it there overnight. And so you get, just a idea of all the sediment and water that comes through. You get a long-term snapshot of what's there. And we found, to my surprise, a large number of Listeria monocytogenes present in those environments. Basically, with close to 3,000 samples tested, we isolated over 1,200 individual strains of Listeria monocytogenes. The diversity of monocytogeny strains we found was quite interesting. We found close to 100 different genomic subtypes. Some of these were subtypes that were very commonly found in foodborne outbreaks, and some less so. But I think the point is that there's a lot driving the ecology of these organisms out there in the environment, where they're naturally living most of their lives. And there are factors including what they might be living on in nature, the decaying plant growth, etc. Also the wildlife that may be carrying them from place to place. And we did find similar strains at areas like 30, 40, 50 miles away from each other. So indicating that the water could carry them or the wildlife could carry them. Just this diverse array of organisms that were there. And we would search for them starting phenotypically. We would do an enrichment culture. I like to do non-selective pre-enrichment cultures because one thing you find when there are so many diverse subtypes of organisms that you're targeting in an enrichment is you get culture bias. So if you have lots of different Listeria in a group, but you only want to find one particular Listeria, like let's say you've got Listeria innocua, Listeria seligeri, Listeria welshimeri, and Listeria monocytogenes in a group, it will tend to happen that the non-pathogens will outgrow the monocytogenes. It's just the way of things. So you try to skew your enrichment media to make it easier for your monocytogenes to prevail. And in doing that, you sometimes feed into diversity within the monocytogenes itself. And so you get some subtypes that grow well and other subtypes that are clamped down on. And so in order to get the broadest snapshot of what's there, I like to do non-selective pre-enrichments and then go further in to look for, with more selective enrichments to narrow it down. Also, especially when you're talking about potential pathogens that you're looking for in nature, it's tough out there. It's tough out there for a bacterium to grow, especially if you're coming from a water source. You don't have a lot of nutrients going around. It's pretty dilute. You're competing with other microbes, and not to mention there's UV, if there's sun around, there's temperature variation, there might be pH variations. The cells that are coming in are not at their best physiological state. They're kind of like how we are at 6 in the morning. They need a little coddling. They need to be not challenged right away, which is, yes, we have coffee in the morning. We've got to provide that for our bacteria to massage them to grow into these more selective media. And so we grow them gently, and then we challenge them. Now, after you amplify the numbers originally, that's when you start using immunomethods, such as they actually make these. They're fascinating things. They make these magnetic beads. They're microscopic beads that are coated with antibodies, and you can they make them with different antibodies, but you can get anti-Listeria antibodies. And so you can take an enrichment culture and mix a portion of that with these magnetic beads, and then you just put the tube up against a magnet, and then you collect those beads and then rinse them off and look for Listeria. And we found those were very good at pulling out Listeria from enrichment cultures. We found that was the predominant method that we use to pull out most of our strains. And then after that, once we got down to the single colony issues, then we would do PCR to verify that we had Listeria monocytogenes.
I think this is so fascinating, and I do want to compliment you on something. And it's this. And this is coming from my entropically challenged, that is to say, old point of view, is that there are a lot of microbiologists today that forget about the organism, and they're more concerned about the DNA per se.
Right.
And that's very powerful to look at DNA, but that DNA is in an organism, and there are aspects of the organism we need to keep in mind. So as you said, kind of a non-selective enrichment to bring up the numbers and then the challenge, this is not how a lot of people think, but it obviously in retrospect is the right way to think. So great.
Yeah, it's a very common way to think when you're talking food safety microbiology, because you are literally looking for a needle in a haystack.
Yeah.
If you're a clinical microbiologist and you have somebody coming in who's sick, you do cultures and you look for bacteria. That's after the person has selected and has amplified the numbers of bacteria that are there. If you're looking for a potential pathogen in a food source, you're looking for maybe 1 or 2 cells in a milieu that contains billions of other bacteria. So that this is a common way that we think in food safety microbiology.
It would be similar to you saying, I'm just going to check people at random to see if they have Listeria in their system.
Right.
It'd be a huge amount of work, as opposed to a situation where people have symptoms and you just look at them.
Right.
And you can't do that with produce.
No.
So I believe you have some beautiful images of both your screen of prevalence of things, and also some attachment images I thought you might want to talk about.
Yeah. So in talking about the diversity of strains that we isolated from our 5-year survey, I do have a phylogenetic tree that shows genomic subtypes, and it's a busy slide, but it's basically just to show that there are about 100 different types. And even though they have different evolutionary relationships to each other, they are all Listeria monocytogenes. And most of the strains actually all of the strains on that survey contain a full complement of virulence genes. And about 50 to 60% contain additional pathogenicity islands, indicating that they might be even hypervirulent is the term that they use. So there's just a lot of diversity that's out there, naturally. And as far as looking at the physiology of Listeria interacting with produce, we did a lot of work with GFP, green fluorescent protein tagged strains. So we have a plasmid that expresses the green fluorescent protein. And then we were able to grow different plants and contaminate them with Listeria, and then just look at the physiology. We wanted to see where Listeria grew and attached to those plants. And so back to sprouts, because sprouts are just a great model system. They're easy to grow in the lab. You can grow them in a couple of days. And what we found for Listeria was that they really seem to attach to the root hairs of these sprouts. We looked at alfalfa sprouts, broccoli sprouts, and turnip sprouts, of all things, but because we just wanted to compare different plants. And I think I have some pictures of that in here. They're some pretty dramatic pictures, just regular stereo microscopy of those sprouts. Root hairs tend to have lots of oxidative compounds in them. There's a lot of reactive oxygenated species that show up around root hairs. And what we found in some of our work was that Listeria strains that had more resistance to oxidative stress had better attachment to sprouts. And so that was one area. And then, but the sprouts are an intact plant, so to speak. But when we're looking at something that might be fresh cut, such as lettuce, we've done some work with GFP in lettuce. And I've got a micrograph of a confocal image of a GFP expressing strain. And what we see Listeria attaching to lettuce, this is, I think it's romaine or it's green leaf, I can't remember which one. It will attach to the cut part of the lettuce, which makes sense because that's where all the nutrients are. So when you tear a lettuce piece, you damage the lettuce cells and you have leakage of nutrients. So Listeria seems to attach to those places and grows. You can also find instances where it will surround stomata inside the lettuce leaf. And so you can find circles of green on that image, as well as there's a broad number of Listeria that are attaching to the big, thick rib inside the lettuce leaf. So there are several places on an intact or on a broken leaf where it can attach and amplify. And if you keep that in your refrigerator, it can even amplify in your refrigerator.
It's so funny. I keep thinking there would be a wonderful time lapse in the refrigerator of that would be a beautiful thing to see.
Yeah, but that would be difficult to set up.
Yeah, especially because the light turns off when you close the door. I understand. But I mean, it's interesting because obviously, if the Listeria is attaching, regardless of mechanism, if it's trying to get food or that there's something about those root hairs that they really like and they're amplifying there, that makes more of the dose that people would be getting when they consume it, which clearly is related to issues of virulence.
Yes, definitely. And don't forget, Listeria lives in nature as a saprophyte. It makes its living out there decaying plant material. So it just makes sense that it would attach to these plants. And a lot of these strains have use plant sugars. There are genes for utilization of plant sugars in them. So genes that, pathways that normally a pseudomonad or another plant more classically thought of as a plant-associated microbe. Those sugars and things that it uses for nutrients. Listeria has those pathways, a lot of them.
I would argue that we're kind of an alternative host, is what you're trying to say.
Yes.
Yeah.
But it seems to have adapted quite well to those alternative hosts. However, the question always is, are you a really good pathogen if you kill your host so quickly? And so I'm actually really fascinated at those virulence, what use those virulence genes have outside of a human host. Since we found so many of those pathogenicity islands in environmental isolates, that suggests that they have a use out there. So is it only in being propagated through wildlife and that cycle and just being reintroduced in different places? That's a question that I think needs some more thought and work put into it.
And something else to consider on that topic, speaking ex cathedra from my navel, is that honestly, if an organism is asymptomatic in you and still uses you as a spreading agent, that's kind of a successful organism.
That's a successful organism.
And there have been cases of that kind of thing. You're thinking of the same person I am. But the fact is, this can happen. And I'm always a little thrown off by the idea of why did evolution do something, because of the Francis Crick quote that was apparently something Leslie Orgel said, is that evolution is smarter than you are. And I think there's some truth to that, but I would not be at all surprised because evolution uses all the tools available. If I understand correctly, there's no evidence of Listeria invading plant cells?
No, we haven't seen any of that.
Interesting.
It can get, well, if it can get into the stoma, potentially it can do it that way. But no, generally I haven't seen any of that, and I don't recall seeing any studies where Listeria was shown to do that.
So fascinating. You know, this has been really a wonderful segment, and I think we've learned a lot from it. I have learned a lot from it. And the first thing that I would ask you at this point as we start to wind up our session is, are there 3 things that you would like listeners and viewers to remember? And probably one of those things should be about food safety.
Yes. We have a very safe food supply. Our food supply is monitored constantly, and the instances of foodborne illness are few and far between. So I don't want to scare anybody away from eating any particular food because of an outbreak that might have happened in the past. The point of surveillance is to find these things and deal with them before it gets...
Earlier in this podcast, we had a wonderful guest talking about broccoli sprouts having all kinds of wonderful probiotic benefits. I guess I'm not using the term correctly. It's not the probiotic. They are feeding the microbiome. There's an expression for that I can't think of right now. But the point is that this guest was really clear that you have to cook these things. So there's no issue with that at all.
Right.
Right. And that's good to keep in mind.
Yes. But there are some foods we traditionally eat raw.
Yes.
But I'm saying you should still eat your vegetables.
That's correct. Because if you are just a carnivore, you have another set of issues to be concerned about.
Exactly.
Yeah. So that's thing number 1.
Thing number 2, and you say this much more eloquently than I do, but this is not our world. We live in a microbes world, and we are just a part of it. And there are no good organisms or bad organisms. It's just nature, and we just adapt to that.
You know what? I had a friend of mine, Caitlin Reese, who's an artist, made an image that I hope gets put up here now. And it's a logo from my class from several years ago. And it shows Frank Sinatra as microbes. And it says, it's not our world, it's a microbial world. And that's a famous story with Dean Martin during the Rat Pack era. Frank Sinatra had done something unusual, and they asked Dean Martin about it. He says, hey, it's Frank's world. We just live in it. And it is a microbial world. We just live in it.
We just live in it.
And number 3?
And microbes are awesomely cool.
No, I mean, seriously, think about the ability to pop a membrane inside an organism that already has an outside membrane, taking advantage of normal mechanisms, and I hate to infer intentionality, subverting them to your own benefit, co-opting the cytoskeleton to move around and avoid the immune system. This is where expressions like hashtag WTM, like, what, the microbe? This is where it comes from.
And actin clouds, they're cool. That's an awesome way to go around inside of a cell.
Yeah, you never would have guessed it. This is what I say is wonderful to be a microbiologist. I'll bet a lot of people are asking this question, Lisa. How do you think we should prepare our uncooked produce?
Here's what I do. I just try to wash everything, and I wipe it with a towel. Like my lettuce, I wash it and I wipe it with a towel. That's not going to take off everything that's there. It's my own peace of mind. And if you can get any bacteria that would be associated with dust or sand that might still, or soil might still be on the produce, it's important to get that off. Not only because you don't want to eat the grimy soil, but any microbes that might be associated with that. That all said, I still eat salads every day. Um, I eat raw carrots and peppers and all of these wonderful things. So
It's one of those issues of convenience versus risk versus, all these different ideas. I remember during the pandemic days, there were people that were trying to spray down their produce, if
right.
you remember, as I do. And I don't think that's reasonable at all. I do admit myself that when I get a bag salad, I still wash it anyway.
I I I am guilty of getting bag salads. Sometimes I just don't have the time, and it's very
Sure.
convenient. And for the most part, it's fine. I don't get sick. We have a very safe food supply.
We do, and it's one of the issues that we don't teach very well is issues of relative risk. We get a
Yes,
little carried away, and again, it's horrible for people that are afflicted, but if those numbers are small, that's very different than a huge number of people.
correct.
Uh, what I'm surprised by the number of people that do a little, grow a few vegetables of their own. They're very thorough about washing those,
Yes,
right?
because they see what goes into it.
And I thought you might take a couple of moments talking about your path in the microbial sciences because I already know it and it's fascinating.
um, I went to grad school at University of Connecticut a long time ago, where I studied soil microbes. I always wanted to study funny microbes, so I was like anything but E. coli because everything was E. coli. Um, so I have a background in studying gliding motility in soil bacteria with both Cytophaga and now Flavobacterium. And then moved on to Myxococcus, where I studied regulation of fruiting body formation in Myxococcus. And then one of the things that always fascinated me, because at the time I went to graduate school, there was a heavy influence. There was a heavy drive to study pathogens and study virulence and study clinical microbiology, and I didn't want to do that.
Mhm.
Um, I wanted to study the weird things that were out there because I thought the microbial world was way too awesome to just pigeonhole it into one little, these are the bad microbes. Um, and I sort of got interested in the question of, well, all these pathogens that are affecting humans are only pathogens for a short amount of time. And most of the time they're out there in nature. So what are they doing when they're not being
Mhm.
pathogens? And I was offered the opportunity to explore that when I was offered a job with the Agricultural Research Service, the research arm of the USDA, to ask just that question, to look for human foodborne pathogens out in nature and try to understand how they became associated with food in the first place. Um, and so I just took my program in that way and looked at the ecology of foodborne pathogens and how they interacted with foods and where they were in nature and how they made their living out there.
So the reason I ask this kind of question is because students often come to me and they think there's only one path to becoming a microbiologist. And you're right. There's that focus on human related issues. And I can roll my eyes about human centric narcissism all the time, right? And it's true, but I'm a microbial centrist, right?
Right?
And that's the way I've always believed. And so the thing that I tell people, and you've done this, is to follow your microbial bliss. And I'm not trying to be silly with it. I mean, you want to find things that genuinely interest you. If you love looking at things that cause disease, that's okay too.
That's
But
great.
you shouldn't be forced into that mold. And that's how it was for a lot of people. I mean, I studied root nodule bacteria, and there were a lot of people telling me not to do that. And I did it anyway. And you can say that things might have gone differently, but I think I have done a lot of things that bring me joy. And
I
you have
have
too.
to. It's
Yeah.
been a very challenging. It's amazing how difficult single-celled organisms can be to a human intellect.
They often appear to resist investigation, don't they?
Oh, yes, they do.
Mm-hmm. Yeah, I just is absolutely true. Lisa, it's been wonderful chatting with you.
It's
And
been great.
Since you're now a relatively close neighbor, I hope we can meet up and I can talk
Definitely.
horror stories and movies with your husband. And we can talk, math, and we can talk microbiology, and I hope that we can stay in touch.
Definitely. That sounds awesome. Thanks so much for having me.
Thank you so much. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with epic links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Lisa Gorski is a consultant on food safety and microbiology living in Olympia, Washington. I'll put a link to Dr. Gorski's LinkedIn page in the show notes. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.